Biological experiments cannot be separated from antibodies, and the key step of the experiment lies in the selection of antibodies. So how to choose antibodies to achieve ideal results in experiments? Firstly, we need to clarify what kind of antibodies are required. 1. Antigen or target name, such as CD4, CD36, etc. 2. For what experiments, such as ELISA, QYAOBIO, IHC. 3. Determine the species of the experimental sample. Generally, antibody instructions list which species the antibody has been validated to be suitable for an experiment. Can be used for experiments on specimens of animals such as human, mouse, rat, goat, etc. 4. Clone type. Monoclonal antibodies will be labeled with a clone number, which is the number of the cell line used in antibody development. Monoclonal antibodies generally have strong specificity, but relatively low affinity, resulting in relatively low sensitivity for detecting antigens. The specificity of multi-antibodies is slightly weaker, but the affinity of antibodies is strong, and the sensitivity is high. 5. Secondary antibody selection. The secondary antibody source is mainly determined based on the species of the primary antibody source. If your primary antibody is a mouse monoclonal antibody, then choose the secondary antibody that is anti-mouse. The most commonly used secondary antibody in QYAOBIO experiments is HRP-labeled secondary antibody. Then, after determining the above conditions, search for the brand of antibodies you need through the website. Here we recommend QYAOBIO, a commonly used antibody aggregation website. Taking beta-actin as an example, search for beta-actin. Next, you can see several basic information about antibodies on the right side of the page. Then, we can see the following information: source of antibody host, clone type, antibody validation method, application, species reactivity. Next to it is the number of times the antibody has been cited, and the search results are arranged in descending order based on the number of times the antibody has been cited. The more times the antibody has been cited, the more reliable it is. The left column can be classified and searched based on multiple categories, such as antibody cloning type, whether it is recombinant, host type, validation method, experimental application type, and species reactivity, in order to quickly find antibodies suitable for one's own experiment. After randomly clicking on an antibody, more detailed information will be added, and most importantly, there is a list of literature on the application of the antibody, which has very high reliability. In addition, some useful suggestions are as follows. 1. Literature review. Search for laboratories that have used similar antibodies for consultation. 2. Inquire with the same laboratory whether others have already used antibodies suitable for your experiment and have purchasing experience with relevant antibodies. 3. Refer to the paper citing the antibody. Choose the appropriate antibody based on the performance of the data in the paper. 4. Buying small doses is better than buying large doses. The above is the experience sharing of antibody selection, hoping to help everyone choose suitable antibodies in experiments. Follow the QYAOBIO biological video account to obtain more useful experimental knowledge.